In this video, we're going to expand upon our knowledge of the structure and chemistry of individual nucleotides and look at the larger scale structures that form when we join many nucleotides together in sequence, forming the molecules that we commonly know as DNA and RNA. We're going to start by looking at DNA first because it has a much more regular and predictable structure than RNA does. In fact, we say that all DNA adopts the almost an identical structure. There are only certain situations, like when DNA is being produced, in which DNA does not form the structure that we see here. So if we look at this molecular structure of DNA, we can see that it forms a distinctive spiral structure and that there are actually two layers of DNA that wrap around each other like this. Now, if you remember from protein structure, you'll remember that a helix is the scientific word for when spiral structures form, but because there are actually two chains of DNA that wrap around each other, we refer to this structure as what we call a double helical or double helix structure, and this generally is the structure that all mature DNA forms, regardless of what organism it is found in. Now, if we look on the inside of the double helix structure, we can find a fairly regular organization like this. So, like proteins, how if we remember what the backbone of the protein structure is, the backbone of the protein structure is the repeating NCC, NCC, NCC chain. And unlike in proteins, however, the DNA backbone is actually found on the outside of the double helix, whereas a polypeptide backbone is actually found on the inside. Now, if we look at the components of the nucleotide that make up the backbone, we see that the phosphate is found most on the outside, with the sugars, the middle of the nucleotide, found kind of on the inside of the backbone, and therefore we call the backbone of both DNA and RNA the sugar phosphate backbone, indicating the different components that make up the backbone. Now, if we look on the inside of the double helix, this is where we can see our nitrogenous bases, and we'll explore this in more detail subsequently, but we can see one superficial detail showing that one purine structure. Remember that adenine is an example of a purine because it has a bicyclic ring structure, forms an attraction with one pyrimidine base, and we can recognize pyrimidine bases by looking for a monocyclic ring structure, of which thymine definitely is. Now, similar to proteins in that proteins also have a backbone, nucleic acids, like proteins, are also not symmetrical molecules in the same way that proteins have the N-terminus as the defined front of a protein and the C-terminus is defined as the back end of the protein. Nucleic acids also have a front and a back, so you can see the right-hand strand is actually written upside down because this is actually the back of that strand, whereas on the left-hand strand, this is actually the front. Now, if we remember how the carbon is numbered, we remember that the nitrogenous base always tells us where carbon 1 is, which means that the phosphate can be found on carbon 5, and this is what we call the 5' end, and again, we can identify where carbon 5 is simply by looking for the phosphate end. So the 5' end, where the phosphate is, or where the first phosphate on the first nucleotide is defined as the front of DNA, whereas if we look at carbon 3, so we'll have to look upside down here, nitrogenous base on carbon 1, there's carbon 2, and here is carbon 3, we would expect that there is an OH group on carbon 3, and this is what we call the 3' end, or simply the OH end, or not OH minus, sorry, because this OH is an alcohol. So the 3' end, where what we are looking at is a terminal, meaning ending OH group, this is defined as the back end of a DNA molecule, and you can see that the 5' end of one strand and the 3' end of the other strand are actually on the same sides, because you would read front to back in the opposite direction on both strands. Now, because the two strands are read in opposite directions, because the front of one end and the back of one end are actually on the same side, we say that DNA is an anti-parallel molecule. So remember from mathematics that the word parallel refers to two lines that will never directly touch one another or never intersect. And this is true, especially if we look at the backbones. The backbone of one end will never touch the backbone of the other end, but we say that DNA is anti-parallel because even though the two strands will never touch each other, they are said to be moving or said to be read in opposite directions. Now, if we take a look at the nitrogenous bases in more detail, we see a pattern. We see that A and T are always found together. So A always pairs with C, whereas G and C are always found together. And this occurs because of what we call complementary hydrogen bonding, and we can actually take a look at this hydrogen bonding here. So ignore the fact that the thymine and cytosine text is backwards. This is necessary in order to show where the hydrogen bonding occurs. So on an amine functional group here, the nitrogen is going to have a negative pole and the hydrogens are going to have a positive pole, which will naturally attract to the negative pole on this double bonded oxygen here on thymine, and likewise this negative pole on this nitrogen is going to form a hydrogen bond with the positive pole on this hydrogen here, whereas if we look at cytosine, we see that this negative pole on an oxygen is not present on cytosine. There's a negative pole on the nitrogen, but there are two hydrogens with positive poles here, which can form a hydrogen bond with this oxygen on guanine, and likewise this hydrogen here will form a hydrogen bond with the negative pole on nitrogen on cytosine, and finally the negative pole on this oxygen of cytosine will form a third hydrogen bond with the positive poles of this hydrogen on guanine like this. So we can see, because these hydrogen bonding groups match each other in what we call complementary base pairs, we say that A and T are complementary because whenever you have an A, it is always complemented by a T because of matching hydrogen bonding groups, and likewise C and G are said to be complementary for the same reason. The final part of this video, we're going to take a look at RNA, which has a few noticeable differences. So remember that one of the skills you'll need is distinguishing between RNA and DNA, and you actually already know two different reasons. Number one, in DNA, we have deoxyribose as the pentose sugar, but in RNA, you would actually find ribose, and if you remember that carbon 2 has an OH group on it in ribose, whereas carbon 2 has only a hydrogen with no oxygen in deoxyribose. This is the same distinction. And then the other distinction to remember is that while DNA has thymine, ribose will instead have uracil as its base. So if you have your data table handy with the distinction between each of the nitrogenous bases, identifying RNA through the presence of uracil is relatively easy to do. Now, the actual differences beyond the pentose sugar and the nitrogenous base between DNA and RNA are actually the main differences are between the large scale structures rather than the individual nucleotides and individual pentose sugars. For example, while DNA only ever exists in its mature form as being double-stranded in the double helix form. RNA molecules, depending on their function, can either be single-stranded, where only a single helix of RNA exists, but you also have many examples where RNA can be double-stranded, and we can see a couple of examples of these below. So unlike DNA that basically exclusively exists in a double helical form, RNA can form a variety of complicated 3D structures, many of which can exist in double-stranded or single-stranded form. In fact, because of its large variety of 3D structures, RNA is often mistaken for a protein. So this form of RNA is what we call tRNA, and we can see that the majority of tRNA is double-stranded, DS meaning double-stranded, but we can see this small section here is a single-stranded form. Now, this complicated structure here that looks very, very much like a protein is a structure that is part of the cellular organelle called ribosomes, and ribosomes actually function as a type of enzyme, but not an enzyme that is made out of protein. Ribosomes, rather, are actually made almost entirely out of RNA, which distinguishes it from the double helix of DNA. Now that we've learned about the small and large-scale structures and distinctions between DNA and RNA, in the next video we'll take a look at what organisms actually use each nucleic acid for.